In this lesson, we are going to explore a very strange phenomena called endocytosis and exocytosis. Now, these are functions of the cell membrane that are used for the bulk transport of particles. Because you see, many substances are just too big to get into or out of the cell by passive or active transport. To absorb large particles, such as bacterial invaders, cells engulf them with their plasma membrane in a process called endocytosis. To export large particles, such as digestive enzymes manufactured for use elsewhere in the body, they often use a process of exocytosis. There are three types of endocytosis. One is phagocytosis, the other is pinocytosis, and the other is receptor-mediated endocytosis. All three involve the basic process of the plasma membrane oozing around an object outside the cell, surrounding it, and forming this little pocket called a vesicle, and then pinching off the vesicle so that it is inside the cell, but separated from the rest of the cell contents. Now, let's talk specifically about phagocytosis and pinocytosis. Relatively large particles are engulfed by cells in a process called phagocytosis, which you see happening here in this image that you see with the yellow background. Amoebas and other unicellular protists, as well as white blood cells, use phagocytosis to consume entire organisms, either as food or as their way of defending against pathogens. Pathogens are disease-causing organisms or substances. Whereas phagocytosis comes from the Greek for the word eat and container, the term pinocytosis comes from the Greek for drink and container, and describes the process of cells taking in dissolved particles and liquids. The two processes are largely the same, except that the vesicles formed during pinocytosis are generally much smaller than those formed during phagocytosis. Well, the third type of endocytosis, which remember is called receptor-mediated endocytosis, is much more specific. It's much more specific than either phagocytosis or pinocytosis. Receptor molecules on the surface of a cell recognize and bind to one specific type of molecule. For one receptor, it might be insulin. For another, it might be cholesterol. Many receptors of the same type are often clustered together in a cell's plasma membrane. When the appropriate molecule binds to each of the receptors' proteins, the membrane begins to fold inward, first forming a little pit and then completely engulfing the molecules, which are still attached to their receptors. One of the most important examples of this receptor-mediated endocytosis involves cholesterol. Now, most cholesterol that circulates in the bloodstream is in the form of particles called low-density lipoproteins, or LDL. You may have heard your parents talking about their LDL and their HDL levels if they're going to see their doctor and having their blood work done. So those are two of the common cholesterols that we look for. Each molecule of LDL is a cholesterol globule coated by phospholipids. Proteins embedded within the LDL's phospholipid coat are recognized by receptor proteins built into the plasma membranes of liver cells. Once bound to the receptors, the LDL molecule is consumed by the cell by endocytosis. Once inside the cell, the cholesterol is broken down and used to make a variety of other useful molecules, such as the hormones estrogen and testosterone. Circulating cholesterol often builds up on the walls of arteries, reducing blood flow and causing the artery to harden. Too much circulating cholesterol in LDL molecules can lead to cardiovascular disease and even death. Individuals lucky enough to have large numbers of LDL receptors on the plasma membranes of their liver cells have a significantly lower risk of cardiovascular disease. On the other hand, some individuals are at risk of early onset of cardiovascular disease because they consume food laden with too much cholesterol, such as egg yolks and cheese and sausages. Or Some have the misfortune of inheriting genes that code for faulty liver cell membranes that have very few LDL receptors. This is a disorder called familial hypercholesterolemia. Say that 3 times fast. In the extreme case where an individual is born with no LDL receptors, circulating cholesterol accumulates in the artery so rapidly that cardiovascular disease begins to develop even before puberty, and death from a heart attack can occur before the age of 30. Let's take a look at exocytosis. So the movement of molecules out of cells takes place throughout the body. For example, cells in the pancreas produce a chemical called insulin that moves throughout the circulatory system. Insulin informs body cells that, there's some glucose in the bloodstream that ought to be taken out and taken in and used for energy. So the insulin molecule is much too large to pass out through the plasma membranes of the cells where it is manufactured. As a result, after molecules of insulin are produced, they are coated with a phospholipid membrane to form a vesicle. The insulin-carrying vesicle then moves through the cytoplasm to the inner surface of the cell's plasma membrane. Once there, the phospholipid membrane surrounding the insulin and the phospholipid membrane of the cell fuse together and ends up dumping the contents of the vesicle out to the outside of the cell where it can enter the bloodstream, which you see illustrated here. Exocytosis is not restricted just to large molecules. In the brain and other parts of the nervous system, for example, communication between cells occurs as one cell releases large numbers of very small molecules called neurotransmitters, and this is also done by exocytosis.